All right. Thanks for inviting me to speak today. And I'm going to be sharing with you work that I've been doing on looking at evolutionary dynamics of human gut microbes. But also I should share a little bit more broadly about who I am. I study evolution in natural populations. And I'm interested in understanding the complexity of evolution when we have changing seasons, many interacting species, spatial structure, and so on. And the microbiome is a really rich place to actually study evolution in a natural population. So a lot of us know that the microbiome is composed of bacteria, fungi, archaea, and viruses. And this taxonomic diversity has been really extensively studied, as well as its functional diversity. But now we actually have a lot of genomic data available to ask also how are these organisms evolving. Now, when we think about evolution of microbes, we can go back to our lessons from experimental evolution in the lab. And there we've actually learned a lot about how bacteria evolve. So the famous Lenski experiments have shown that E. coli, for example, can evolve really rapidly and repeatedly. And so I'm showing that here with an adaptive mutation arising in a population and rising to high frequency. So the question is, does this picture of rapid and repeatable evolution extend to the microbiome? Here you might actually see something quite different given that you have such a complex community, whereas in the Lenski experiments you are studying one species at a time. And so instead of evolutionary processes, I'm actually missing a slide, I just realized, but instead of evolutionary processes you might be having ecological shifts in species composition, or you might be having invasion of a better fit strain from the outside environment, or you might actually see evolution where you have a genetic modification that then propagates through its subpopulation. And when I'm talking about evolution, I'm talking about a change in allele frequency and not a change in species composition. And I'm specifically meaning a change in allele frequency that another change can then propagate on top of in the future. So I'm not speaking about, let's say, a very closely related strain that comes in and then goes out. I'm talking about really a genetic modification that occurs that another one can occur on top of later on the same haplotype background. Now, there's actually abundant opportunity for evolution in the microbiome. There's a really nice calculation done in Zhao and Lieberman's paper where they take the rough estimate of number of microbial cells inside of us, which is about half of the cells in our body, combine that with the mutation rate and generation time and genome size, and they come up with an estimate of about 1 billion mutations entering our microbiomes daily. And so this is just a really rich and fertile ground for evolution, and yet we don't really quite understand how it proceeds in this complex environment. And I think we're really at the beginning of that, and there's a lot to be discovered. So what happens to these billion mutations that enter our microbiomes every day? Well, actually, the processes that determine its fate are population genetic processes of drift and migration, adaptation, and recombination. And these processes have been extensively studied in all sorts of populations, but not so much in the microbiome. And we really should care about this for studies in the microbiome because the gradual loss or fixation of new mutations can actually confer a variety of traits that really matter a lot for our health. So, for example, to illustrate why genetic variation in our microbiomes matter for us personally, one example comes from this paper where they found a genetic variant in a population of Japanese individuals that helps to digest seaweed. And it came from actually marine bacteria. Another example, which we're all very familiar with, is antibiotic resistance. And again, the genes inside our microbiomes play a really key role. And then finally, we have drug metabolism. And this is a really cool story where there's a species called Egretella lenta that some of its strains can actually inactivate this drug called digoxin. The mere presence of Egretella lenta is not really a marker of inactivation of the drug, but it's the variant inside of it that matters. So, and how did all these genetic variants come to be? Well, through the population genetic processes of drift, mutation, migration, adaptation, and so on. And another reason to study evolution in the microbiome is that there's a very a tight connection, actually, probably with ecology. And for me to actually draw this distinction of, does the microbiome respond only by these ecological shifts of species compositions or by evolutionary changes, I think is actually false. I think given the rapidity on which these two processes are co-occurring on the same timescales, these are probably inextricably linked, and they're probably influencing each other. The ecology of the system may likely modulate the evolution of a focal species and vice versa, and the microbiome is just a really wonderful place to study this. So recently I wrote a review on population genetics in the microbiome with Katie Pollard, and so today I'm going to be giving you a bit of an overview of what I think is the state of the field and what some of the open questions are. I'll be highlighting my own work, but also sharing tidbits of other prominent work that has recently come out in the field. So before I delve into each of these different processes, I'm going to first actually talk about data, because that's actually what we need to make our inferences from. So, okay, what kind of data do we need to study population genetics in the microbiome? Well, a lot of people have been using 16S data, and this is a really fantastic kind of data to profile which species are there. But as many of you know, it serves to barcode which species are there and doesn't really tell you much about what else is going on in the rest of the genome. And so this actually means that we don't have the polymorphism information that we need to actually detect evolution. And so a different kind of data that we could use is shotgun sequencing data, whereby we sample everything from our sample. These are all the genomes, and then we fragment them into small pieces and sequence them. And as you probably know, this is a really challenging type of data set to work with because the reads are only 100 base pairs long, and it's this really complex community that's all mushed together. The field is really advancing, and we are starting to get data that contains linkage on longer length scales. So, for example, we have isolate sequencing, single-cell sequencing, metagenome assembled genomes, and to just highlight some really recent advances in the field, there are 3 efforts in concert. I'm only highlighting 2 of them here that actually pooled a lot of metagenomic data from public sources, and were able to identify just a tremendous number of new taxa that we'd never been able to see before due to culturing abilities. So our abilities to actually see what's in our microbiomes is actually really expanding. Now, I'm actually going to be showing you work that I've done with shotgun data, because this is actually really ubiquitous, and I'm going to show you some methodology that I and my collaborators developed to track evolution using this complex type of data. So what we do is we actually map reads. We try to simplify this data. So our goal is to try to understand which sites in the genome might be evolving new mutations. And this is like searching for a needle in a haystack, but what we can do is just first, right off the bat, just try to simplify the data. And so we can do that by mapping reads to different reference genomes. And as you just saw, our reference genomes are now really greatly expanding. And then we can go in and identify polymorphisms, such as here, where we have 1 at 50%. And so once we do that, then we can go ahead and look at genetic diversity across a bunch of samples. So for example, let's say we look at this blue species. Let's say it's Bacteroides vulgatus, because it's very common in the population. And we can look at a panel of adults and see what its frequency is, what this allele's frequency is at this particular position. And we can see that it really greatly varies, indicating this is probably a very interesting site because it's so polymorphic, both within and between hosts. Now, a lot of people have been actually using this kind of information to track transmissions of strains from host to host or one environment to another. And so we might see there's been efforts whereby, including my own, where we've seen transmission of strains from moms to infants. And people have done that by looking for marker alleles that are present at high frequency in mom and baby only and are totally absent in the broader population. And so this type of analyses have revealed that although the species level diversity starts to resemble mom with time in the baby, marker allele sharing actually decreases with time. And so looking only at the species level information hides a lot of important information that tells you about modes and mechanisms of transmission. So, all right, let's now talk about drift and migration. I'm going to be just going through each of these different topics one by one. And given that you now have a sense of the data, we can ask the question of, is everything everywhere? This is a really popular question. And so one way we can start to answer this is actually by looking at ourselves. Well, how many strains of a given species colonize a host? So like, let's say again, we're talking about Bacteroides vulgatus. Do we expect exactly one strain, a few, or hundreds? And so back to our cartoon of how to look at this genetic variation, what we can do is actually look at the genetic variation within each host to try to see which of these different models is consistent with the diversity we see inside of each host. And so what I'm going to do is I'm actually going to draw a histogram of the allele frequencies for each of these hosts. And so let's take this given host, and I'm showing it actually for 3 different individuals from the Human Microbiome Project. And again, I'm showing it for Bacteroides vulgatus. On the x-axis, we have allele frequencies ranging from 0 to 50%, and then on the y-axis, the fraction of sites with that allele frequency. And so what you can just see right off the bat is that there are these really funny bumps, and so you can conclude that there's just a lot of variation across hosts. So what exactly is generating these modes and peaks in the data? So it turns out, as you can see, we have a peak at 0% and then, or close to 0, and then at around 15%. This peak here is actually not expected under one cohesive population, a panmictic population. Instead, what this is representing is a mixture of 2 populations, here what I've shown is a blue and a green strain, a blue and a red strain that are mixing, and these mutations that arose long before the 2 strains colonized the host are what's generating this peak. And why 15%? Well, it's because one strain is at 15% and the other is at 85%. So it's that mixture and the long evolutionary timescales that separated these 2 strains that's generating this peak. And so you might be able to then guess what's going on in the second host. You can see that we have a shift to 50%. What's causing that? So here, again, we have a colonization by 2 common strains, and one is at blue and red, and again, here they're separated, they're present at 50-50 frequency rather than 15 and 85. Now this guy has a much more complex situation, and we actually cannot conclude whether this person is being colonized by 3, 4, or some other discrete number of strains. But we can say that it is discrete because you see these modes. So basically, if we were being colonized by exactly 1 strain or hundreds of strains, we wouldn't see these bumps. And so basically, we can say we are oligo-colonized by a few strains. Now the question is why? What are these processes? What's the underlying ecological reason or evolutionary reason? And that's something to be determined. Also, some species actually, only exactly one colonizes the host, and I've highlighted a paper here which goes into depth as to why. So within us, it doesn't seem like everything just promiscuously gets in, but does that have implications for global population structure? Is everything everywhere, or is there some structure around the globe as well? And so looking at gene content, here I've highlighted a recent paper by Pasoli et al. You can see that actually there is different genetic compositions in non-westernized and westernized populations, and you can see this in different ways around the world. You can see also at the SNP level, this is from Churung et al., that we also have a lot of biogeography. Interestingly, not every single species has biogeography. So in work that me and my co-author did, we found that for Bacteroides vulgaris and for other species, we actually have very closely related strains found on totally different continents. In China and US, they're not identical because they do have some genetic differences, but they're extremely similar. And so while you have a lack of structure around the world for some species, you also have these very intriguing patterns of similarity in different hosts. Yeah?
You try to see for the variants within the human gut, because we know that the microbiome differs between different sites within across the gastrointestinal tract, right? So maybe these variations are, and in the stool we see everything, so maybe just where it all flushes.
So are you saying that we have different variants along the gut?
I'm asking, I'm not saying.
Oh, sorry. Just trying to make sure I understand the question. So that you have different variants along the gut, and that maybe it's a sampling issue?
Yeah.
Possible. I don't know. And it could be, but the question is, why do you see such similar strains on different continents? Maybe they're identical. And so we see this for many species. I'm just showing you one example here. And the question is, maybe it just so happened that we got unlucky and there happened to be a person sitting from one data set, HMP, sitting next to another person from the other data set on a flight, from Chin et al., and they happened to exchange their microbiomes. And when we checked in the data to see if there was this consistent overlap for all the shared strains, it was totally random. It was basically like looking at a null distribution. So it's kind of very mysterious as to what are the factors that result in similar or same strains in different people. So is everything everywhere? No. And there's a lot to unpack in the future. As I mentioned, what are the ecological and evolutionary processes that govern this oligocolonization of individuals? And why do different species show different geographic distributions? Is it a property of the species, of the host, or maybe some interaction between the two? Okay, so let's talk about adaptation. And I have actually highlighted a bunch of papers on the bottom here that are concurrent with mine. I think it's a really exciting area of research that people are delving into in different ways, using different types of data and different types of approaches. And so I'm going to share what we did, which was to look for really rapid allele frequency changes within hosts on short timescales. What we were really happy to see is that other people were also seeing similar things. And the punchline is that there's a lot more to be discovered. So, as I mentioned to you, we want to be able to look for evolutionary dynamics using whole genome shotgun sequencing. And we wanted to be able to do that by looking at these allele frequency shifts over time from low to high. So here is an example where you have a mutation that goes from 0.15 to 100%. Now, this is actually not as simple as it looks. And actually, Murat really highlighted this really well in his talk, where he said, well, look, you might have different populations. And so this is actually a really dicey thing to just say that you're looking at SNPs changing frequencies. And so Murat took a different approach by looking at the amino acids, but what we did was we actually tried to look at what the population structure was inside of hosts. And so one really, just to illustrate this further, a really big issue is that it may not just be evolution that's driving allele frequency changes, but it could actually be shifts in the strain composition. So the blue strain, for example, could go from low to high over time. And that could make it look like you have an allele frequency change due to evolution, when really it's just some ecological shift. So to illustrate why this matters and why we should care about these strain shifts, if you were to grab a strain of Bacteroides vulgaris from you and from me, or any of us at random in this room, on average, they are diverged by 1%. So 1 out of 100 base pairs is different. Whereas what we expect to see in this evolutionary changes over short periods of time are just a handful of differences. Not 1 out of 100 base pairs changing, but maybe 1 in a million. So, that's just a really different order of magnitude. So what we want to try to do is to disentangle allele frequency changes due to strain fluctuations versus evolution. And the complication is that all we see with our eyes in the data are allele frequencies. We don't actually see the underlying distribution of the populations. So going back to our plots where we're looking at the population structure within hosts, you've already seen what the structures are. This person's colonized by 2 strains and at 15% and 85% and so on. Now, what we want to do is, and we're going to exclude the complicated individual communities, because what we want to do is actually resolve a haplotype corresponding to our most common strain. And that then allows us to confidently see an allele frequency change due to evolution rather than a strain shift. And so basically what we have are 2 sites. We can look at a toy example where you might have an allele at 15% and at another at 85%. And basically it's easy to see that you can basically assign the dominant allele to the dominant strain, thereby phasing its genome. And so this is actually a very complex sample to work with, because you might have allele frequencies at 50-50 frequency, and it's easy to misassign the wrong allele to the wrong strain. So basically what we want are samples in which we have a very clear dominant strain in which we can assign an allele very confidently to the most common strain in the sample. And that way we can then go ahead and look for allele frequency changes on the background of that dominant strain over time. And that's going to be our signal of evolution. So you might be asking, well, how many of these special samples do we have in which we actually see this dominant strain present? And it turns out that we actually have a tremendous amount of data. We lucked out in the fact that we are oligo-colonized, and you can see that across hosts, so the depth of each dark blue bar is the number of samples, and across the most prevalent species, we actually have 801 resident populations to work with. So that's just a lot of data, and what's really nice in the microbiome field is that we can start to do population genetics in these different species in parallel and try to understand differences and similarities in evolutionary patterns and rates. But for now, actually, what I'm going to be doing is I'm going to be aggregating the data across all these individuals and species, because that gives me more power to be able to really tell you more generically what's happening evolutionarily and how. So we've done all this hard work now for actually phasing the most dominant lineage, and now what we're going to do is go ahead and take a look at these allele frequency changes going from low to high over time. And so what I'm going to do is I'm going to plot that distribution for our 801 samples on a survival plot. So on the x-axis, I'm showing the number of SNP changes. On the y-axis, I'm showing the fraction of comparisons with that many SNP changes or more. And this is what the data looks like. And so you have this very interesting distribution where actually on the first axis, or first point on the plot, indicates that you actually have 0 changes in 88% of your population. So this isn't news. It was actually shown that in general our microbiomes are pretty stable over short periods of time. But what is news, given our ability to phase the most dominant strain, is that 9% showed a handful of differences of about 5 to 10 SNP changes. So this actually hadn't been seen before, especially from complex shotgun metagenomic data. And we had to be really sure that these aren't false positives, and there's actually a lot of bioinformatics under the hood ensuring that we don't have mismapped reads and so on, and we have an error model under there. So every SNP change that we see, we're extremely sure of. And then you can see that 3% are actually showing a lot of changes on the order of 10,000. So it's a really weird multimodal distribution where you have either very few SNP changes or you have a lot of SNP changes. So why would you have just a few and why would you have a lot? It turns out that there are actually two processes that are generating this. So you could have invasions or you could have evolution. So how does that work? So invasions, you would have, but when I'm talking about evolution, I'm talking about a handful of differences that arise over time, whereas invasions, you'd have this very long evolutionary time that would have elapsed that would have generated, on average, 1 out of 100 SNP differences. And so that actually, in a genome of size, a megabase, results in about 10,000 SNP differences. And in fact, if we were to compute, on average, the number of SNP differences within hosts versus between hosts, you can see that within hosts it's on just a few hundred SNP differences, whereas between hosts it's on average 10,000. So just to reiterate, I'm talking about differences in evolutionary time. And what we can do is use what's going on between us as a control for understanding what's happening inside of us.
Nandita?
Yeah, please.
That's not, you just showed the plot. That's not due to transfer of a genomic island or horizontal gene transfer. Right?
Oh,
You know, the health base pairs coming in that's brand new.
I see what you're saying.
Or even carrying in an allele between diversified.
Yeah, I don't think that this is, we do actually see evidence for horizontal gene transfer, which I'm going to get to at the end, but I don't think it's due to the entire, it could be that we have an entire genome swapping out, and what we're left with is just a little bit of the original one. However, that would then indicate that we have a totally different strain present.
Well, they'd also be linked.
They would be linked,
right.
but then the original guy would have disappeared. So we're talking about like a massive genetic change. So there definitely is recombination happening inside of us, but to result in 10,000 SNP changes, you're talking about like a huge amount of our genomes swapping out. And maybe recombination played a role in that, but then you have to ask what happened to the original genome.
Yeah, so it's something that's diversified to start with.
Exactly. So, okay, so we already saw this data, and now what we want to do is try to understand, is this evolution, is this invasions? And what I'm going to do is I'm going to draw a line for what's going on between hosts. And so here it is in red. And you can see that it's a very different shape. And actually what we have are most of the differences being at around 10,000. So that allows us to classify what's going on inside of us as either evolution or invasions. And so this was really exciting because it meant that, yes, our bacteria inside of us can evolve really rapidly, and that actually has a lot of potential for being able to respond to rapidly fluctuating selective pressures like changes in diet or antibiotics or other health issues and so on. And so even if our own genomes cannot change rapidly and compensate for or adapt to changing environments, our metagenomes might be able to help. So what happens over our lifetimes? What I've just shown you is what happens on 6-month timescales. But the question is, you know, we see a little bit of some small fraction of our population changing over in 6 months. Does that change turnover keep happening such that after 20 years, every strain gets replaced? Or is it that some hosts are extra susceptible? Maybe some population has antibiotics and that results in a big turnover. Or is it that our strains continually to become well adapted to us such that replacements become less likely with age? So they keep accumulating genetic adaptations and such that it becomes virtually impossible for a new one to enter the population. So we can go ahead and try to answer this question. And the ideal kind of data that we'd have to answer this would be longitudinal data over a few decades. And so this is obviously kind of difficult to collect, though actually there was a great paper that was recently published by Yaffe and Relman in which they actually had samples from a single individual over 10 years. But in this situation, we didn't have that. And so we can instead leverage twin data. And so Corpella et al. Showed that cohabiting teenage twins and siblings share many strains. And so there happened to be another data set by Shi et al., which sampled adult twins that grew up in the same house and then lived apart for 40 years. And so what we asked was, do adult twins share the same strains or not? If they do, then it means that they stick around with us for a long period of time. But if they don't, that means that there's a lot of turnover over our lifetimes, and there could be something in between. So going back to our plot, I'm now going to add another line to this plot in showing what happens when we compare twins. And here it is. You can see that it overlaps what's happening between hosts almost exactly. And so this indicates actually that after 40 years of living apart, these strains are completely turned over. And so it seems that replacements dominate over our lifetimes. Now, why is that? You know, there's actually a lot of open questions here. I'm only listing just a few of them, but there's just a lot to unpack. We've just started to understand that there is evolution going on inside of us, but how does it happen? So, and what's the mode and tempo of that? So to just read off a few questions that I think are exciting avenues for future research include, does rapid evolution impact the ecology of the microbiome? Right now, I haven't really even shown you how that impacts the ecosystem and vice versa. How does adaptation impact host health? How much adaptation is there? Right now, we were extremely conservative in how we looked for adaptation because we didn't want to confuse this with sampling error. But if we had more higher resolution data, we might be able to uncover many more events. And what are the targets of selection? We suffer from annotation issues, and so if we had that, we might be able to paint a better picture as to functionally what is adapting. And then also regarding this replacement question, why do they happen? Is it happening in bursts or gradually? And so understanding more exactly how and why adaptation, evolution, and replacements all happen is I think, going to be an area of exciting future research. So finally, I'm just going to lightly touch on this last but very important process in natural populations, which is recombination. And this is a very important force because it can actually play a big role in maintaining a cohesive population. And so there's this really seminal paper from 1993 by John Maynard Smith in which he drew this picture of different levels of recombining populations. So in this instance, you just have a clonal population where you don't have any recombination. In this case, you might have recombination within a certain clade. And then in this case, you might just have a totally panmictic population where everything is recombining with everything else. And so the question is, how clonal are bacteria? And we can try to answer this with the microbiome. And so just out of interest of time, I just am painting a very broad picture of what's going on, but there's a lot of detail that I'm not really providing here. But basically, there are different modes of horizontal gene transfer. This is just textbooky stuff like transduction, conjugation, competence. And then you have many modes of incorporation. So you can have homologous recombination, non-homologous recombination, and so on, in which actually the donor DNA gets incorporated into the recipient DNA. And a lot of people think that there isn't much recombination happening in bacteria because they reproduce asexually. But actually, people are finding that this does actually occur quite commonly inside of us and also across us. And so tools for doing that include using statistics like the 4-gamete test, in which you're looking for all 4 combinations of pairs of alleles on 2 different sites, which would indicate that you have evidence for recombination. And then also linkage disequilibrium is a really powerful tool in which if we see a decay of it over distance, we know that we have evidence for recombination. And indeed, we saw in our paper that we have this LD decay across a panel of hosts, indicating that recombination is not uncommon in the natural population of bacteria residing within us. And more specifically, I wanted to just share with you that recombination might be playing a really important role in seeding the evolutionary events inside of us. So one could ask, are the mutations that are rising to high frequency de novo ones arising spontaneously, or is it that we have some uptake of genetic material that's already pre-existing in the broader population? And so to answer that question, what we can do is take a look at the properties of the mutations that are rising to high frequency in this part of the plot. And what we can do is we can compare it with an expectation under when you have only de novo mutations rising to high frequency. And so first, what I'm showing you are the number of SNP changes that are happening, and I'm plotting their prevalence in the broader population. So our null model is that you would actually have very low prevalence. If this is all de novo, it should just arise at really rare sites. But actually what you can see is that there's 50% of our data arising that's rare and 50% that's actually very common. And so that suggests that what's evolving, the sites that are evolving inside of us actually are polymorphic in other people too. We can also look at the properties of the SNP changes, whether they are synonymous or non-synonymous. And under a de novo expectation, you'd have a lot of non-synonymous sites changing, such that you'd have a DNDS of about 1. And instead, actually, what we see in the data is that DNDS is lower than 1. We actually have fewer non-synonymous sites changing than we expect by chance, and more synonymous sites changing than we expect by chance. And so what this means is that purifying selection has had a chance to operate on the mutations that are rising to high frequency, which means that they're not just arising yesterday. They arose some time ago, had chance to sit around in the population, let purifying selection purge the bad stuff out, and then went to high frequency. So this is not new variation necessarily that's rising to high frequency. And then if you don't believe me, we actually also saw gene changes over time. And this is actually becoming a very common observation in the literature, in which we saw both losses and gains. And the gains have to happen from somewhere. They can't happen spontaneously. And so putting these 3 pieces of evidence together suggested that recombination seeded sweeps actually were potentially playing an important role in evolution inside of us. But again, there's a lot of open questions. Do bacteria recombine randomly with each other? There's some evidence to suggest that there are some intriguing patterns suggesting that the recombination in these organisms is not the same as in like flies or humans. There's actually somewhat of a non-randomness to it. Maybe the oligocolonization plays a role in that. And when do they have the opportunity to recombine if hosts are oligocolonized? They have to see another strain to recombine. And how does recombination impact the mode of adaptation? We talked about that really briefly, but a really big question in the field that has interested scientists for a long time is whether we see gene-specific sweeps or genome-wide sweeps, and so on. And that can have really important implications for local versus global genetic diversity. So, to conclude, what I spoke about today was population genetics in the microbiome. Genetic variation in the microbiome really impacts our health. There's a lot of data to work with. There's a lot of intra and inter-host population structure. Bacteria can evolve in the human microbiome on short time scales. There's extensive recombination. And the rapid evolution in a complex community affords us the opportunity to study eco-evo relationships. I'm recruiting for my lab, so if you know of anybody interested in this topic, please let them know about me. And I'd like to thank Benjamin Good, who's my co-first author on much of the work that I presented today. It's published in PLOS Biology, so check it out if you're interested. Katie Pollard, also an author on this work, and also a co-author on the review we wrote together, and Oscar Halachek, who is an author on our PLOS Bio paper. And so I'd be happy to take questions and stay in touch.